in order to fight against various diseases and in order to treat various disorders. And similarly, there is a repurposing purpose, means the existing drug may be used for other purposes. So it is a wonderful opportunity to have such kind of webinars for the benefit of students. And on this occasion, I wish to extend my sincere thanks to the office bearers, governing body members, in particular QSAR Garu, for giving a MOU in between Bapatla College of Pharmacy and Cepra Labs. I am very much thankful to the managing director of Cepra Labs, Sachinarayan Garu, and the colleagues of Cepra Labs for arranging such kind of seminars for the benefit of our student community. Thank you, madam. Now I request Srimati Sushma to introduce today's resource person. Thank you.
Thank you, sir. Myself, Sushma Arlagadda, an assistant professor from Department of Pharmaceutical Chemistry and Analysis in Bapatla College of Pharmacy. On behalf of our management, Bapatla Education Society, our governing body members, principal, sir, staff, and students of our college, we cordially welcome you all to this webinar. It is now my great pleasure to introduce Dr. Sai Murali RS, who is our subhand speaker, on the topic drug designing and development, a closer walkthrough in silico approaches and prospects for young professionals. Dr. Sai Murali RS holds PhD in Biosciences from Sri Sathya Sai Institute of Higher Learning in 2014 on the thesis entitled Biological, Biotechnological, Molecular Docking, Spectroscopic and In Vitro Functional Studies on Vaccine and Total Alkaloid Extract from Arthoda vascanis. Today, this task of introducing our guest of honor and speaker is a great privilege. I know this one is difficult, but the thought that he is a man of virtue and simplicity, I feel elated to introduce him to everyone at this moment. After all, he is a living inspiration to the young ones all over here. If one takes a closer look at the alchemy of the achieving person, two distinct virtues pop up. Besides perseverance and hard work, these are pioneering spirit and willingness. Our guest of honor and speaker today is a living idiom of a person who happened to be born at the right time and at the right place, coupled with his impressive background. And after his professional outlook, Dr. Sai Murali RS is having more than 15 years of experience in analytical field. Dynamic professional with over 10 years of experience in teaching, research and development, and 5 years in research administration. And adept with proven leadership and strategic planning capabilities led to the development of 4 centers of excellence in LPU in remarkable that is in 12 months of time. He is also having excellent communication and whole grip on comprehension abilities like pedagogy for 14 life sciences courses for 10 years and higher education curriculum development for 10 years and academic board meetings for 5 years and research advisory board for 5 years. And he is also having the capability of building anti-development towards common goals. Coming to the research and development, he is having a grip on plant biotechnology, animal biotechnology, bioinformatics and analytics, drug receptor interaction, phytochemistry and environmental bioremediation. Coming to the professional qualifications, he started his career in the Department of Biosciences in Sri Sathya Sai Institute of Higher Learning. He taught postgraduate and undergraduate students in various life sciences and biotech subjects, adept at international administrative tasks that are performed with the department and institute. He is having greenhouse management, and then after he moved on to the Division of Research and Development in Lovely Professional University from January 2016 to till date. Our expert taught 4 life sciences courses for postgraduate students. He is having an administrative research coordinator for 4 diverse schools, that is School of Bioengineering and Biosciences, School of Agriculture, School of Fashion Designing, and School of Hotel Management. Member of Research Review Board for screening proposals of the faculty members towards external funding. Our expert is having research policy making and execution at university level. He is the head of Central Instrumentation Facility, established CIS that comprise state-of-the-art of instrumentation such as FESCM, powder XRT, GCMS, MSFID, FTIR spectrophotometer, DSC, TGA, fluorescent spectrophotometer, electrochemical workstation, etc. He is also having Center of Excellence. I already mentioned you that he established 4 centers of excellence creating knowledge hub in partnership. That is, he established 4 centers in Brooker LPU Center for Excellence for Microstructural Studies, Shimaju LPU Center for Advanced Chromatography and Mass Spectrophotometer, Geol LPU Center of Excellence for Advanced Microscopic Studies, Perkin Elmer LPU Center of Excellence for Material Characterization. And when we move on to the major outcomes of doctoral research of our speaker, selection of allied chemical types for cultivation of farmers, and plant tissue culture protocol optimization for Arthropoda vasica, drug repurposing, that is identification of novel therapeutic properties of vaccine as potential drug for anti-cancer drug against breast cancer, and mechanistic aspects like theoretical molecular docking and experimental results, topoisomerases, and double-stranded DNA as potential interactors of vaccine. He is also having grip on method development of vaccine using LCMS or SMS. Coming to the technical expertise of our speaker, he is having a hand on bioinformatics, that is, he is proficient in bioinformatics software, and also coming to bioanalytics, he is skilled in using UV visible, FTIR, fluorescence, PCR, DCS techniques, chromatographic and mass spectrophotometric techniques, proficient in analyzing secondary metabolites. He possesses good experimental, analytical, and data interpretation skills. He is also trained on FESCM, XRT, TGA, and electrochemical workstation. And he is also having a hand on biotechnological department. That is, he is expert in plant and animal kingdom. That is, coming to the former positions of our speaker, he is worked as a head for the Central Instrumentation Facility, Division of Research and Development in Lovely Professional University, Punjab. And also, he worked as a full-time teaching faculty in the Department of Biosciences for Sri Satya Sai Institute of Higher Learning. And currently, our speaker is working as a Senior Executive Level 1 Planning and RA for Cipro Labs Limited, Sanat Nagar in Hyderabad. Coming to on an overall view, coming to the core competencies of our speaker, he is having a hand on research administration and research development, team management, and strategic planning. Coming to the teaching and training experience of our speaker, he is having content and curriculum development and research advisory board. He is good at policy making for design structural competent. And at the high end which we need, he is having a hand on instrumentation facility management and centers of excellence management. So right now we are very happy to have you here, sir, as a resource person, as a staff hand on all the fields. Thank you, sir, for accepting our request. And finally, that is, I thank our body member for giving this such an opportunity. Sai, sir, you can share your screen and start your presentation.
Okay, thank you so much, Dr. Sushma, for the introductory note, and I feel very much humbled to be part of this webinar program and being associated with very good teachers from Bapatla College of Pharmacy. Now, coming to the topic today, I'm going to talk about drug designing and development, a closer walk through in silico approaches and prospects for young professionals. So I'm not going to talk anything very exhaustive, but very simple and a story of a drug molecule, how it starts its life, okay, and how it reaches the society. So this is what I'm going to talk, and what are the roles and responsibilities of the students, and what are the prospects that they have in various fields. This is what we are going to talk. That's the subject matter for this webinar. So the outline is molecular docking. I'll be briefly talking about molecular docking, what exactly is it, and QSAR, qualitative structure activity relationships. These two topics from the in silico approaches I'm going to extract and try to figure out how I can present to you in a meaningful way, and drug discovery pipeline in general, and career options for prospective pharma professionals. So if you have any queries in the middle also, you can stop me, and you can send a message, and I can take your voice and try to respond immediately. Even otherwise, in the end, I can figure out how I can help you out with a productive answer. So molecular docking, the goal of molecular docking is identifying key nodes in a complex network of genes, proteins, and small metabolites which are legitimate drug targets in varied proportions. So first of all, what is a drug molecule and what is a target? This is what we need to understand. So if suppose a particular disease is evident, and we need to get some drug candidates out, then the very first thing which comes to any student's mind is that how best a small molecule can be used to target a particular disease condition. So in a disease condition, you will find various enzymes, right? Various metabolic pathways, and various types of proteins which are intermediates, and you also find various small molecules which are secondary metabolites, right? So these all things are to be addressed. So how best we can design a particular chemical to approach a target is what we intend in molecular docking. How a particular small molecule is going to go and sit on a particular site of a target molecule, like macromolecules, like proteins, nucleic acids, or lipids. So how these are going to affect them is the subject matter. So I'll be simplifying the entire scheme into 3 steps. The first one is the virtual screening and docking. So what is in silico actually? Anything that we do using a computer is called in silico. In silico means the silicon, the chips on which we are going to run our algorithms and trying to get some results out of it. So this is what we call as in silico. And virtual screening is nothing but screening various drug molecules on a in silico platform. So that's what is virtual screening is all about. And docking is also conducted in a virtual environment. So it's a molecular virtual environment. So nothing you need to do on bench. So this is called as a theoretical model. So we are going to speculate, or we are going to predict, or we are going to find out what is the probability of a particular small molecule to interact with a particular target, whether it's a macromolecule or even with other small molecules. So here in the first step, we are going to select a chunk of various drug molecules, prospective drug molecules, which we call as small molecules or ligands. And these molecules, we are trying to figure out how the structure or the ligand-based properties can help us develop the molecule for approaching a particular target or the protein. So in this particular thing, the second step is the visualization. Once you dock a particular molecule, the small molecule, the ligand and the target complex, how they are interacting, okay? and how they are aligned. This we can visualize, and visualization actually helps us even as the undergrad second year or so. We used to get to know how the stereochemistry works and how the molecules are aligned, and teacher shows us with the model in the hands. So similarly, visualization here helps us to understand how a particular drug is interacting with the target. And then the third step is very crucial for understanding intricate things of how the efficacy of the drug could be and things like that. So the bonding pattern and the hydrophobic interactions which are there with the target protein. So in general, the scheme of docking is, the first step is selection of receptors. So receptors are nothing but the targets, and the selection of interactors. Interactors are nothing but the ligands which we are designing as prospective drug molecules. Now the receptors are the ones very important, and we need to identify that based on the literature survey. Like what exactly are these receptors, what are their functions in a particular metabolic pathway, and how they are playing a crucial role in a particular disease condition. So these are the important things which we need to know about the receptors before even taking receptors. Example, I'll give you one example. I worked on topoisomerase. You all know about topoisomerase. It's a DNA unbinding enzyme which is important for relaxing the DNA double helix during the time of replication, right? So if suppose we are targeting DNA topoisomerase, we are designing certain molecules which can inhibit DNA topoisomerase, and further by inhibiting DNA topoisomerase, we are going to stall the replication network as such. And the replication, once it is stalled, the cell will stop growing or the dividing, right? So that's one of the potential areas where we can target cancers. So that's one of the areas. So the topoisomerases are of different types. Like TOP2 alpha is there in humans, and TOP2 beta is also in humans. Similarly, we have topo 1 and topo 2 like that in the bacterial counterparts, and these we call as orthologs. So you have a human protein or enzyme, you also find the same thing in a bacteria. Okay, so there is certain things which are very, very important for life as such, and they are retained over evolution. So these topoisomerases are very legitimate targets, for stalling a particular cell growth. And these orthologs can be targeted. So selection of receptor is very important. We can find out what are the orthologs of that particular receptor. Example, topoisomerase. We have different orthologs. In bacteria, you find topoisomerases. You find that in Caenorhabditis. You find it in Drosophila. You find it in Arabidopsis. You find it in humans. So it's common in every other organism. But what is very specific is how much of it is conserved. The sequences are conserved. Active site sequences are conserved. And how much of it is varying. Okay, if suppose the active sites are conserved, then we can design molecules which can inhibit even bacterial topoisomerases, which can inhibit even human topoisomerases, and we can have both antibacterial action and also anti-cancerous action simultaneously. So selection of receptors are very important, and once you select receptor, then the second step is preparation of receptors. We need to prepare the receptors. Now, selection of receptors, where do you get these receptors? The structures, the chemical structures of these receptors. You can get the 3D structures from PDB, a database which is very renowned one now. It is Protein Data Bank, and you can get all the extracrystallographic structures of these receptors. And what you need to understand here is you need to get a very good resolution X-ray image of this particular receptor. So when you're getting, you can't get just like that. you have to get specific high resolution X-ray structures of these receptors. And once you have these receptors, you need to prepare the receptors. Why you need to prepare the receptors? Because you need to remove selectively certain contaminants or inorganic contaminants or water molecules which are accumulated during the process of crystallography. And once you remove these contaminants, then your receptors will be free of other inhibiting factors, and these receptors can now interact with the ligands which you are developing. Now these receptors, once they are prepared, okay, you have different softwares to prepare these receptors, like you can even do it in PEB itself, and you can also use PyMol. You can use PyMol, a software where you can prepare these receptors. By using this, you can remove water molecules, contaminants, and you can also do the energy minimization. And that's very important, because once you remove certain contaminants and water molecules, the receptor will have certain areas where it's not having the right energy frequencies. The bonding pattern and things like that, everything will change. So if you minimize the energy, it will come back to its originality, and it's now ready for accepting the ligands for binding. Similarly, on the other hand, the interactors should be selected. Suppose say you have you have an alkaloid, just like vesicin or even certain, even if you take Taxol, you have the structure of the Taxol, and this Taxol, you can find out what are the similar ligands which are having same backbone like Taxol, and then take those structures and try to figure out how they also can be contestants in this particular drug design. So preparing ligands is also equally important, just like preparing receptors. And here in ligand preparation, we are going to again use a different software called MOE or something like that, where you can energy minimize the ligands as well, and then make sure that the ligands are clean and they are taken from right sources. So PubChem is one source where you can get these ligands. That's the site database, and you can also get from ChemSpider. You can get from Zinc, very open access databases these are. And once you have the ligands, you can also search for similar ligands in the same databases. And once you have these ligands as grouped ligands, you can take them as the training set and then start docking. So the next process is, once you have the receptors and the ligands, you have to make a grid. You have to arrange a grid. What is a grid? A grid is nothing but, suppose this is the fist, and I don't need the entire fist to be docked with the ligand. I just need certain portions of the fist where the ligand could probably bind, and those portions I need to identify through literature review. I need to identify through removing selectively the receptor ligand crystallographic structures from where the receptor is actually acquired. So when we are removing the unnecessary ligands which are already crystallized with the receptors, that specific portion could also serve as a source for knowing where the interactors can actually bind. So once you have the grid assigned, our ligands can specifically be docked in that particular location or around that location, and figure out what is the binding score. So binding score is nothing but what is the best possible stereochemistry in which the ligand is binding to the protein molecule. Okay, protein is a macromolecule. It's having a quaternary structure, right? So in a three-dimensional structure, how this particular ligand is going to bind to the target molecule. That's what we are going to analyze in the binding score analysis. So once you have the binding score is also called as binding affinity of the ligand to the receptor. Then you will visualize on a Lick plot where it's an interaction diagram. And then you can also find out that the other things like the bonding pattern and hydrophobic interactions. So this is the grid assignment where the specific portion of the protein molecule you will locate. And here 3 important things you need to understand. One is the grid coordinates. What are the grid coordinates? The X, Y, Z axis, the points, and also the grid centers, how actually they are oriented. So you can move the grid as the box which you are seeing over there. You can move the box according to your usage. Okay, you can reduce the box size. Okay, you can increase the box size, but by increasing the box size, you are giving more possibilities for the ligand to dock, to be docked at different places, right? There could be more than one place where the ligand can bind. So you may get unnecessary binding patterns, which you can eliminate by rationally designing a particular spot, okay, based on your literature review and also theoretical understanding. You can locate and put this grid at the point where you need to bind the molecule. So once you bind the molecule, okay, once you dock the molecule, you have the receptor and ligand complex, okay, and once this complex is formed, you get a binding score and the possible pose in which the particular compound is interacting with the target. So here binding score is given in minus 8.7 or whatever it is. This is nothing but a free energy. It's a binding free energy, and this is counted in kilocalories per mole. So once you dock the molecule with the target molecule, the small molecule with the target molecule, you will find interestingly an interaction map. Okay, this you can get in PyMol, which is another software which you can use with your docking software. So for docking, you have one software. You have PyMol or visualization tools which can help with the docking software together. Nowadays, you have Autodoc Vina, which is Scripps Institute's one, and it's freely available. It's an open source one. You all can try. I used to play with that software when I was a student, PG student, and after that I gained grip over that and developed my own docking platform. So here, interaction analysis, you have certain molecules which are called the amino acids in the proteins and how they are oriented around the ligand, or in other words, how ligand is interacting with different amino acids in a protein, okay, and the best possible conformation or the orientation of the pores you can determine this. And you can also find what is the type of interaction it is having. If you see in this two-dimensional map, the link plot, okay, you might have seen this in the biochemistry textbooks, okay. Here you will find these are the moieties, the amino acid moieties, which are there all around, methionine, serine, like that. So many tyrosine, things like that. So how this molecule, the drug molecule, is interacting with the target protein, okay? At what residues or what amino acids it is interacting, what kind of interaction it is. See, this is a hydrophobic interaction which this molecule is having with the target protein. Similarly, you can have two different docking poses which can give two different angles of viewing the same interaction in a two-dimensional plot. So here also you will find different types of bonding patterns. See, here you will find a direct bonding pattern where even the bond length is also given, right? So an interaction tool will help you understand how the molecule is aligned and how it is interacting, what kind of interaction it is having, whether it's a hydrophobic interaction, electrostatic interaction, whether it is a hydrogen bond interaction, okay, and what is the type of bonding, what kind of bond length it is having. All the details you can figure out from the docking poses. And the importance of visualization can be best understood. If you see here in this particular diagram, you will find a drug molecule going into the cleft of a protein and sitting over there. Okay, you can identify certain pockets in the protein where exactly this drug molecule is going and fitting. Okay, if you recall your enzyme kinetics or enzyme chemistry, you will find out how best the docking or the induced fit models can be understood. Similarly, here you can understand how the ligand is going and selectively docking, getting docked at certain portions of the protein. So by visually understanding, you can figure out how you can redesign your molecule, what analogs you can design as a medicinal chemist. So visual examination of predicted binding geometries contribute crucially to the further development of a lead compound, either towards enhanced binding affinity, towards reduced side effects, or towards reduced susceptibility to drug resistance related to mutations. So in virtual screening, so we have discussed about the molecular docking as such. Okay, what is the general process, how we select the receptors, how we select the ligands, what is the grid position, why grid position is important, and once it is docked, what is the score that is a readable thing for you to understand what's the affinity of the ligand to the target molecule, and what's the interaction type, right? So part and parcel of this is the virtual screening. So it goes all the time throughout the docking procedure, where you will understand that this particular structure of a chemical, okay, or a small molecule, is similar to other molecules in the library, compound library. So there are tens and thousands of molecules out there in the library. You can figure out structurally how they're related, and by taking this, you will term it as structure-based drug designing. Okay, so there are different types of drug designing. One is structure-based drug designing. The second one is lead-based drug design. Other one is pharmacophore-based drug design. I'm not going to confuse with all that, but try to understand that the first and foremost thing is very simple to understand. One is that structure-based drug design, because you're all chemists and pharmacists. Now, if you see the virtual screening, so why you need a virtual screening? Because manually we can't select, we don't have experimental setup to conduct various assays. We don't have enough resources or the funding options for supporting our experiments, and we don't want to unnecessarily waste our chemicals on certain procedures where it's not required actually. End of the day, if it is not going to hit that market, then it's of no use, right? So by using virtual screening, we can select tens and thousands of molecules, and see here 5 lakh chemical compounds are selected in the beginning, and by using different filters, we landed up getting around 100 compounds. So these 100 compounds after molecular docking, okay, only 30 compounds could be tested in experimental setup. Only 30 or even less than that could be tested in experimental setup. So your costs are reduced by this rational drug design, right? So you don't need to unnecessarily pump your funding into wasteful endpoints. Then the second topic is quantitative structure-activity relationship. Okay, so we discussed about the drug designing part using one particular thing, that is the structure-based drug designing. Okay, now we are moving on to the other one, which is a quantitative structure-activity relationship. There is a common thing in between these two things, is the structure. Okay, so structure is common between these two methods, but here we are going to figure out how a particular compound is having a particular function. So you are going to assign a functionality to a compound, and based on the functionality, you will interpret whether it's good to take this compound further for commercial purpose, or to detain it, or to modify it. Several strategies you can plan accordingly. So structure is directly related to the function that we understood. So modification of structure could possibly lead to change in the property, that is figurative. Then structure modification can can be done in two ways. One is intentional structure modification through in vitro projects or virtual in silico operations. So among these two, the second one is much rational because even the FDA and ICH guidelines state that we should have the QSAR based approach at one point or the other during the drug development pathway. So virtual screening in silico operations, we are taking QSAR as the source, and we are going to use the workflow of QSAR to address this particular predicament. So chemical space to property space modeling could be done in QSAR, and receptor independent versus receptor dependent processes are available in the QSAR platform, which you can explore further. So in general, the scheme of QSAR is you have electronic databases, okay, which is stored in the public domain and also in very specific domains. And then lab collections, you have different experiments which were done earlier in labs, and these experimental results are also out there in the public domain, and you can use some zinc, PubChem, and ChemSpider, these type of databases to know what are the functional annotations of a particular chemical compound. Okay, so this you can find out in zinc. I prefer zinc because you get lots of other parallel databases in zinc where you can access even the pathways. You can have the holistic understanding about how a metabolite is getting affected. Then we have the literature data, which can also serve as the starting point for the data collection. So once the data is collected, and it should be curated because we don't want false negative ones, so or false positive ones. So we want exact data. So we have to curate the data and remove unnecessary things. I'll tell you how we curate the data. Then we have the data repository. Now the repository could be taken for the QSAR Workflow platform. So once the QSAR Workflow platform is on, the software is on, okay, it's 5 minutes job. But then before doing that and after doing this operation, you will have to work a lot. So doing QSAR is not a big deal, but understanding the process by which a particular molecule is pumped in, and pumped out of the QSAR platform is very important, because after the QSAR platform, you need to give your interpretation, and that's very important to understand how a particular molecule is going to affect an endpoint. So here QSAR works after the models are generated, they can be then taken to virtual screening and molecular design. Okay, just like how I discussed with you earlier in the first part. You can also go for experimental validation if suppose the assays are very, very feasible. Now the chemical library could be selected, so you can see a big chunk of chemical library is taken, and this particular chunk is now pumped into this screening process using empirical rules, chemical similarity filters, QSAR based filters, feasibility or availability filters. So end of the day, you will get only few potential hits or leads. The first part of the predictive QSAR workflow, the actual software of QSAR, is curation, data curation. So the chemical data set which you get, you have to curate it, and here in the curation of the database data set, you remove the mixtures, inorganics, and other contaminants which can potentially affect your workflow. And then the structural conversions or cleaning or removal of salts and normalization or the energy minimization, then the treatment of the tautomeric forms and the manual inspection is very important. So these are the things which you use in the curation of the data set. So there are prefixed system criteria which you will use in this particular process, and after that you will generate the descriptors. So descriptors are nothing but the ligands where we are talking in the first stage of drug designing, similar to the ligands there. here we call them as descriptors. So once the descriptors are there, these descriptors can be taken further through machine learning techniques, and further they will be having the data repository back of the software, and how best these are aligning and giving us a meaningful information. So end of the QSAR workflow, you will understand a particular descriptor, how it is having a particular functionality, or how it is affecting a particular process in the human physiology. And thereby you can deduce some inference stating that that particular compound is good or bad for a particular situation. So descriptor types, you have several types of descriptors. One is the constitutional, then topological, geometrical, thermodynamic, and electronic. So different types based on the type of information that these descriptors can give us. So first one is the constitutional one. It's very simple and commonly used descriptors reflecting the chemical information of a molecule without any information of atom connectivity. Okay. The second one is the topological one, where the connectivity of the atoms in a molecule, small molecule, could be figured out, and this will lead to pharmacokinetic properties and etc. etc. Then you have the geometrical ones, where you get the three-dimensional coordinates of the atoms, and you will understand how they are interacting with the target moiety, and that's the protein, and you will figure out a whole lot of things there, by interactions. Then the thermodynamic ones are the ones which are related to the structure or the chemical behavior of the molecule, and that's again the traditional way. And then you will also find out the molar refractivity and things like that in this. Then you have the electronic ones, where you will also understand the dipole moments and things like that, and you will try to figure out how that is going to affect our whole lot of functionalities. Then prediction of biological activities. If I take you back to the literature, 1863, you have the pharmacological activity was directly understood as a function of concentration by Crum and Brown, and that was the starting point of QSAR. And then linear relationships between lipophilicity. So you all know about the lipophilicity, how important it is for absorption of a particular drug, right? So even the Lipinski's rule of 5, you might be knowing about this. It's very important for the absorptibility of the drug molecule into the body, and the small molecule should not have more than 5 hydrogen donors, and it should not accept more than 10 hydrogen acceptors, and then it should not have more than 500 kilodaltons of molecular weight, and it should not have more than 5 of the partition coefficient, right? So these are the 5 principles of the Lipinski's rule of 5, which are taught over a period of time in the drug absorptibility, and that's very important even here to understand the QSAR, because functions are related to the absorption. Then Fulner's geometrical activities also are important, because increase in the geometric progression also will lead to increase in the availability or decrease in the availability of the drug molecule. So QSAR tools are used in the absence of available data for prioritization, classification, screening, level of risk, and in the risk assessment as such. That systematic and consistent approach, this is the one which is used even by FDA, and ICH guidelines strictly suggest us to implement QSAR platforms to understand the drug functionality. So the endpoints related to related to solubility, hydrophobicity, absorptivity, and volatilization. These are the endpoints which we are actually thinking, which are very simple to understand and also simple to operate on a QSAR platform as well, because you have a lot of literature already accumulated on these parameters. Unlike the biological processes which involve the kinetic, enzyme kinetics or physiological interactions in the pathways, metabolic pathways, because they involve a lot of organ systems and it's very complex to understand. Right? For that, we need experimental models to substantiate the theoretical understanding. So now for these four types of things, solubility, etc., we have the softwares which are having inbuilt mechanism to understand the physicochemical properties of chemicals, and they give us more rational outlook of how they are going to have a particular function. So advantages of QSAR models, you can reduce the costs, you can reduce the time, you can reduce concerns related to conducting toxicity bioassays. Input data requirements are generally modest and increasingly available through public and computer-based databases. There's a distinction between SAR, that is structure activity relationship, and QSAR, quantitative structure activity relationship. The only thing is, here in QSAR, you will try on a series of structurally similar chemicals, and you will try to quantify them. Okay, and here you will use lots of statistical tools like regression and things like that, or the decision trees, or the neural networks, etc., to discriminate between the right endpoint and the right functionality. Then applications of chemical screening, prioritization, regulatory and corporate decision making. How QSAR is important for this particular aspect. So nowadays, ICH guidelines, the Q7, etc., even Q3, they state that how your compound is going to damage the genetic composition of humans. Okay, how it is going to have an effect on environment, ecology. So these are the things which we need to understand, and that could be understood using QSAR platforms. Then so regulatory users, if you see, they support priority setting of chemicals, guiding experimental design of regulatory tests or testing strategies, then providing mechanistic information, then grouping of chemicals into categories based on similarity, then filling a data gap needed for classification and labeling, and filling a data gap needed for risk assessment. When it comes to the reliability and validity of QSAR, no software-based technique can give you 100% reliable or valid information. Okay, so always experimental data is important, and in the absence of experimental data, then only we are going for QSAR or software-based techniques or computer-based techniques. So the first point is you need to understand this is a predictive one. It's not exact or it's not empirical. Okay, so it only gives you a clue about how to navigate your process. It won't give you a definitive endpoint. Okay, so you need to figure out how experimentally you can support this particular QSAR data. So reliability of QSAR estimates vary with applications and endpoints. Environmental fate endpoints, for these endpoints, relatively the results are well accepted because it's not going to have a fatal thing on the environment as such. A small molecule cannot harm that much fatally an environment unless it is an ethylium bromide. So human endpoints are very important for us for discussion. So non-mammalian toxicity endpoints are more reliable as high quality data is already available, and we can use the QSAR platform using that particular data, whether this particular compound is having a toxic effect or not, we can understand that. and very few legal regulations are available or ethical concerns are mitigated, because you can perform non-mammalian studies easily, like on insects, etc. You can always perform studies and find out how a particular molecule is toxic. If it is toxic to Drosophila, definitely it might be toxic to other animals as well, right? So lots of studies were already done on Drosophila, understanding the toxicity of compounds and taking it further. So there are very few legal implications or concerns related to these studies. Then empirical data is greater, it serves greater reliability than the QSAR predictions. So the bottom line here is collective evidence of the empirical data, and QSAR will give us best possible source for judgment. Now, human health endpoints are mutagenicity, carcinogenicity, teratogenicity, neurotoxicity, reproductive and developmental effects, and then skin or eye sensitization or irritation, and then systemic toxicity. So these are the ones which are targeted generally in QSAR platforms. So these are some of the open source softwares which you can download in your laptop itself, and then try how you can start learning docking. Okay, so for docking you have various things like Autodock. I suggest Autodock is the simplest one and very efficient one. Autodock Vina is the latest one which you can use, and visualization tools like Rasmol, PyMol, etc. you can use. And then you have energy minimization tools, which I told during the first slide, like the preparation of ligands and targets. So you can use various softwares here. You can use MOE also, which is a molecular operating environment, which is user-friendly one. It's a paid version, but then you have QSAR descriptors, then chemical drawing and software libraries. So these are the softwares which are open source ones. Now coming to QSAR as such, you have paid ones like Open Practical Knowledge Acquisition Toolkit like TopCat, then you have Multicase, then Derek, then PBT. These are the ones which are used for various purposes, very specific purpose. Some are very selective to environment, some are very selective to toxicity. So based on that, you can select your package as such. Then other solutions are like Oncologic and various platforms are available online, which you can explore. Then supporting programs and packages, you need certain conversions, like one file format to other file format you need to convert, okay, during the docking process or during the energy minimization process or so on and so forth. So that time you will use softwares like OpenLabel, etc., which are very useful for converting from one format to other format. Then replacement therapy. So we discussed about the docking, we discussed about QSAR, how these in silico tools are used in rational drug designing. Okay. Now coming to next part, that is the drug development. So though drug designing is part and parcel of drug development is very specific to pharmacy students, and that's the reason why I got these topics into the picture. Now here, replacement therapy is one area which is gaining a lot of importance these days with the onset of biologics. Okay, here if suppose a particular protein or an enzyme is defective, okay, which is very important in metabolizing a particular drug molecule. Okay, if it doesn't metabolize that particular drug molecule, then the effect is nil. Okay, you may get a 100% result in vitro studies using a particular drug molecule, but in vivo models fail. Why is it so? It's because of non-availability of the drug to the respective target, or the drug is not getting metabolized in the first place. So protein replacement therapies are used in line with the drug designing, okay, or the mainstream drug development, and that can be further taken forward. If suppose a drug is promising in vitro and no activity in animal model, again, the delivery systems or the formulations should be targeted. So here two problems could be faced. One is the chemical doesn't even get into the body, and the other one is the it fails to reach the site of action. Okay, so these are the two possible problems which you will encounter, and that's because effective treatment might require brief application with rapid reversals of effects. Okay, and then the second one is the drug delivery may be required over months, and in some cases like hormone therapies, you need to give pulsated drug delivery in small intervals. Okay, short intervals you need to give pulsated drug delivery. Then targeted drug delivery, example in cancers, which has very specific patterns. They show you can't have a general anti-cancer drug just like that given to control a particular cancer type. So specific cancer chemotherapy requires a targeted approach. So in general, from bench to bedside, the journey or the fate of a drug molecule you can find here. The first and foremost step is understanding the underlying molecular, biochemical, and genetic mechanisms, and trying to integrate that with bioinformatics and genomics knowledge using in silico platforms, and then trying to figure out how you can possibly get a potential drug target with critical role in a disease condition. Okay, and then how chemists or medicinal chemists can help design some compounds which can be potential targets. Drug candidates, and then the drug development pipeline as such. If you see, it's a complex evaluation process. Fraction of the preclinical phase the compounds can survive. Only fraction of the compounds can survive the preclinical phase, and then only few go all the way to final approval by FDA. So this is very crucial for us, as students, as teachers, as industrialists. We need to understand how this process can be improved. Okay, and as of now, this process is very cumbersome, and there is no straightforward pathway for this. We need to undergo lots of mitigations. We need to undergo a thorough screening process. So the drug candidates which are coming out of this box, the drug development is very, very few. They are very few. So the career options, you have multitude of career options in pharma. Okay, and the whole world was shut down. Okay, only pharma company and the Medicare were active. You can understand the role of pharma in the society. And the whole world was shut down. Okay, when teachers were not even getting their pay, okay, only the pharma industry survived. So you have a lot of possibilities. You have a very, very committed role in the society, and people respect you for that. You may not understand at this point, but when you see from the other side, you understand lots of things, how government looks at pharma, how industries look at pharma, how people look at pharma. It's mind-boggling. It's just amazing. And we have specific roles in each and every thing. Okay, so you have various types of branches in pharma. One is the pharmaceutics, where you design a particular drug. You try to figure out how the drug could be taken forward in the drug development pipeline. That can come under pharmaceutics. The subject matter, what we discussed so far, comes under that. Then the pharmacokinetics, how the body is affecting the drug molecule. Okay, whether the drug is getting absorbed or not, whether the body is facilitating the drug absorption or not. That you can figure out from pharmacokinetics. And pharmacodynamics, how the drug molecule is affecting the body, right? So that you can figure out from pharmacodynamics. And nowadays you have pharmacokinetics and dynamics together, PK/PD, There are various mathematical models which are available, which you can use in the drug development pipeline. Then pharmacology, you all know the in vivo studies, how important they are and how beneficial they are in the drug development. And then pharmacogenetics is nothing but, if suppose a particular gene is responsible for a particular protein, okay, and that particular protein is very important or crucial for metabolizing a particular drug molecule, and if suppose that gene is getting mutated, okay, that is the subject matter for pharmacogenetics, and your role there is to figure out how and what kind of mutation has happened and how we can replace that particular gene and things like that in the therapeutic systems. Then the pharmacogenomics is nothing but, if huge number of such genes you are taking, then it's called as pharmacogenomics, and you use a lot of computational biology also in this. We call it as system biology. Then regulatory, you know, ICH and other guidance and FDA, guidance documents, they eloquently speak about, you know, how the drug should be getting to the market. So we need to undergo very, very stringent measures, and regulatory is very, very important. It's the backbone of industries. Any drug which is coming out is because of regulatory scrutiny, right? Unless that is done, it cannot come out. Then the last one is clinical sciences, okay? How in clinical setup you can find out a successful in vivo drug can behave and how better you can get it out into the market. So these are the ones which are there. Apart from this, you have even the traditional type of the scientific approach, where you can become scientists and you can do PhDs in relative areas, and you can specialize and you can become teachers who can train many, many students and guide their futures. Now, that's one way. That's anyhow there on the table. Other than that, what are the things you can do? You can go into industries. You can go and work as analysts. You can go and work as QA personnels. You can go and work as QC personnels, right? With proper understanding about those areas. You can also work in regulatory sciences. These are the additional things. Apart from the bench work, you can also go into the government agencies, into the patent offices for patent law, and investment firms for investment evaluations, and you can go into consultancy. You can go for editorial roles in journals or publishers places, and you can also go into establishment of your own firm, being an entrepreneur, right? So these are the things which are out there, and you can figure out how best you can use your thinking process. Don't be biased. That's a sincere suggestion, because people will be biased generally. If one of our friends he or she is doing a particular course or going into a particular area of career, we try to mold ourselves into that area. That's not right. Whatever you are best in, okay, you should figure out how you can go into that particular area. And if you see the previous slide, here you will find different branches of science, okay, of pharmacy are interrelated. They are not in one angle, right? They have multiple phases, and you can go into different branches using the same different knowledge of various branches. You can figure out how you can address a particular problem in that particular branch as well. Like example, I never thought that drug designing can help me in the industry. Okay, I never thought that it would help me in industry, but now I'm in regulatory affairs, and drug designing is very important for us to understand how better we can serve the regulatory affairs. Okay, how better we can screen the compounds and give a green signal to the compounds for the markets. So having said that, you have wide career options out there, and apart from this, you can also have your own setup, your own firms. You can have your own startups, and that's very important. Government is also looking forward for encouraging such young entrepreneurs. And with this, I think come to close. Students' perspective. if you see, we are mostly biased, so we need to get out of that. We need to open up our mind, okay, and try to learn as much as you can at this stage, okay, so that you can go into various realms and work using other realms as well. The knowledge of other realms can never go waste. It will come in hand, and you can figure out how you can apply that in your own field where you're working. So timely discovery and development of new medications are very important. So whenever you talk about drug molecule, then definitely molecular designing or molecular docking will come into picture for any learned person. So always try to learn this particular software-based techniques so that it will help you in some way or other in the future. Then from small pockets of lab research to large interdisciplinary groups working together, we need to expand our scope. So it's not important to work in a particular area and do our own research and publish a particular thing. It's important to collaborate with others and try to learn from them as well, and also give our learning to them, and together we'll grow. And that's the philosophy of modern day drug design. And industrial requirements. When it comes to industrial requirements, there's huge potential for pharmaceutical students or pharmacy students in the industries, and you can explore lots of opportunities in the pharma companies. Then global challenges, we have pandemics hitting, and I don't need to talk anything about it because you all know what's the devastation that COVID actually caused to the entire world. And we have responsibility, on the other hand, to tackle such situations, and pharma and Medicare people are the ones which are first sought after. So in summary, we discussed about the molecular docking and QSAR workflows as part of the in silico techniques, just to understand at a basic level how these techniques can help us in the drug design and development pipeline. Then in silico skills are important for us in multitude of career opportunities, and not restricted only to a particular field or dimension in the pharma industry. So these are selected references which I used for this PowerPoint. So thank you so much.
Hello. Hello. I think there is some technical issue because there are some other queries from YouTube. So meanwhile, you can answer them, I think so.
Okay.
So the first query is, can you suggest some tips in selection of mobile phase?
For example, salt is soluble in water. So you are anyway.
Voice is not clear, sir.
Thank you, sir. Thank you, Simon, sir, for your valuable explanation about these two topics. So I request our participants, that is our students and staff, if they are having any queries regarding these topics, they will be asking the questions and you can just clarify them.
Good afternoon, sir. Shall I ask questions, sir?
Okay, ma'am, you can carry on.
Sir, valuable session, sir, from your side. Sir, according to me, we are familiar with the only Lipinski rule of 5 only. The selection of ligands which obey the Lipinski rule of 5. With respect to other than the oral absorption, the molecular weight will be less than or equal to 500. What about the if molecular weight is above the 500? Sir? How we are going to select? Sir? Sir, am I audible, sir?
Yeah, you are audible, ma'am. I got your point. So first thing is, we'll try to avoid having more molecular weight in the first place. Okay, that's the first thing. It works a lot because molecules out there, and you can screen a whole lot of but if suppose there is a potential problem in some molecular weight and that molecule seems to be a very good contestant, then definitely we have to take it forward. Then we have to understand what is the functionality of that molecule as a whole. Like what are the various functional groups which are associated with it, and how important are each functional group in a particular mechanism, right? So we are now going into mechanistic way, right? This is an alternative way where if you don't have a molecular weight, the criteria, it's not meeting the criterion of the Lipinski's rule of 5, then we'll have to redesign the molecule as such. We have to understand its functionality, the functional groups, how important they are in a particular function, and try to avoid those functional groups and reduce the burden on the molecule first thing, or try to figure out if there are other molecules with the same backbone, okay, and if they can be used as substitutes than this molecule. That's the rational way or legitimate way of looking out for other molecules. But general, we have to avoid such molecules because we know the problem is there with the molecular weight. Why we need to take it up further?
From my side, sir, one more question. Okay, sir. One more question from my side, sir. For the selection of targets, whenever we are taking from PDB, there are so many isoforms are there, sir. Which one is good for docking, sir? And how we are going to run the isoforms, sir? Is there any criteria, sir, for selection of ligand target?
Suppose a particular already available in the database. You have to understand what kind of molecule is bound. Suppose it is a topoisomerase. Okay, if it is bound to DNA, then definitely we know that it is bound to DNA. Okay, and along with DNA, if it is bound to some inhibitors, which are DNA binding inhibitors. Okay, then we know that there is a keen or intricate relationship between these three, right? So we have to identify such molecules which are important in line with our research. If we are looking out for DNA interacting compounds, okay, which go and bind specifically to DNA, like ethylium bromide, a good intercalator, okay, or etoposide, which goes and binds to DNA and also to the topoisomerase, okay. So such molecules, if they are already there in the crystal structures, if they are bound with the target molecule, that is the topoisomerase and this molecule, if they are bound together and there is a complex, okay, such crystal structures we can explore, we can shortlist. And very important thing in this is the resolution of the crystal. That's very important. The more resolved the crystal structure is, the better it is for us, for our studies. So the Angstrom should be always less. If it is 1.4, then if you get 1.21 also, then you have to go for 1.2 because that's more resolved and good for our study.
Thank you, sir.
Good afternoon, sir. So my query is that can all these approaches can be applicable for even marine drugs and herbal drugs too?
So whenever we are trying to figure out a small molecule, how it is affecting a target molecule, okay, we are not talking about extracts. We are talking about molecules as such. So they are chemicals. They are not extracts. They are not compounds. So in Ayurveda and other fields, other traditional fields, where they use extracts, they act in a different way. Physiologically, they act in a different way. Sometimes they are they have much more potential than the isolated compounds. So we have to understand how these compounds are acting along with others. So that's the synergistic effect, which we call. That's a different subject as such. But as such, we are taking the chemicals. We are not going into the extracts. We can't drop extracts. Right?
So this is not possible to go through for all this.
That is much more beneficial to the current context because the individual compounds are failing, but the extracts are outperforming in some cases. So now the traditional approaches are focused. There are lots of journals, foreign journals, where they talk about alternate or complementary approaches for drug design. So I think future is a little vague in that case.
Myself, Nagamonika, of 4x4 B Pharmacy. So, after the generation of receptor in PDB format, we will prepare the receptor. So, while preparing the receptor, the process of preparation of receptor is same for all the receptors, or it may differ from one receptor to another receptor.
All the receptors will have same process, but when you are doing the energy minimization, okay, different force fields are used based on the chemistry of the molecule. Okay, so theoretical understanding is important for that particular stage, where you will specifically use a particular force field for energy minimization, because we need to understand the stereochemistry or the structure as such, the quaternary structure or the tertiary structure as such, and then possibly give solutions for that energy minimization.
During the preparation of receptors, we will eliminate the unwanted substances. So some receptors are macro. So different chains may be present. So how we will eliminate the particular chain that is the unwanted? How can figure it out like it is unwanted chain?
You know what is the composition? Which are there in a DNA molecule. The nucleotides, basis, whatever. We know about them. Now, in crystallography, we use solvents, solvent molecules, which we know that they are solvents and they are not part of DNA. They go and bind to DNA just for the process of crystallization. Okay, so during the process of crystallization, these molecules sometimes they get settled in the structure itself. So we need to eliminate that, and crystallography will not give us solution for that. What we need to do is we need to in silico remove those molecules, and computer does that for us. There's an algorithm which actually removes those compounds. So we don't need to invest any other thought process on that. The computer does that. If we ask it to remove the water molecules, it identifies the water molecules and removes the water molecules. We don't need to do anything.
Thank you, sir.
Sir, I'm Padma Priya. Sir, in silico approaches, up to how much extent we can predict safety efficacy without testing the in vivo or in vitro studies?
That's a good question. There is no definitive answer, but more or less around 90% we can predict. 90%, but that is not a as such. So, more than 97%, and that's not possible in silico. So around 90% we can predict whether a particular molecule is going to have a definitive functionality or not. But based on QSAR data, definitely we can predict much more than that, because there are already existing compounds for which there is experimental data, okay, and the QSAR predictions are also available, and we can map our compound of interest. If suppose it is having similar structure, okay, with slight variations, we can map this compound with that and find out how much, what degree of efficacy this compound also will have in the real life situations.
Thank you, sir.
Sir, I am Madhavi from 4x4 B Pharmacy. Sir, my question is, what is the percentage of drug from drug development to drug approval?
Our knowledge, it's very less, very less. If suppose you have 10 to the power of 8, 10 to the power of 8 is the size of your compound library, okay? The amount which is going to come is only in a handful compounds, like something like 6 to 7 compounds or even less than that, the potential leads, which can stand all the scrutiny. So only 5 or 6, and that's the reason why even the drug development companies are struggling a lot these days. Even top level companies like Novartis or Ranbaxy or whatever it is, Pfizer, they're all struggling because, you know, though they have sufficient intermediate leads, they are not getting the final leads because they are failing at one point or the other in the drug development pipeline, which I mentioned to you on the right hand side of the slide.
Thank you, sir.
Is any other questions?
Sir, 10 to the power of 8 అని చెప్తున్నారు కదా, అంటే is it 1 million for one drug?
Suppose they have 5 lakh end of the pipeline, you will only get 7 to 8 compounds filtered. Frankly speaking, you will get FDA approval only to 7 to 8 compounds, not more than that. Even less than that you will get, because their scrutiny is so very stringent, very, very stringent. That's the reason why you get only few molecules. So 5 lakh to 7 compounds. You can understand the level of because compounds came out only because of your hard work.
Thank you, sir, for giving valuable answers to all the questions. Sushma ma'am, please carry.
Thank you, sir. Thank you, sir, for your valuable explanation on these both two topics. And I hope all our students were spec and span with these two topics. And as you have shared your PPT with me, I'll be sharing those to my students and staff. And if they are having any doubts, as you told that you'll be free to share if any doubt, we'll be pleased to ask you if any doubts we are having. So on this issue, I would like to thank our management body members for giving us such an opportunity to share such an expert ideas with us. And I would like to thank especially Dr. Kishore Babu Garu, that is Ramneni Kishore Babu Garu, for giving us such an expert with us for clarifying our topics which we have not have in-depth knowledge of this. Thank you for giving this opportunity. Thank you, sir.